Hello everyone, thanks for that iMeta giving us the opportunity to introduce our research. The paper name is Microorganisms as Biofilters to Mitigate Greenhouse Gas Emissions from High-Altitude Permafrost revealed by nanopore-based metagenomics. About 25% of the Earth's terrestrial surface is covered by permafrost. China has the third largest permafrost area in the world, and most permafrost in China is controlled by elevation and is called high-altitude permafrost. However, the microbial responses associated with greenhouse gas production in thawing permafrost remains obscured. Here, we investigated the microbiome in the active layer of permafrost, at the Qilian Mountain in the Tibetan Plateau, using the ONT-based metagenomic and Illumina-based RNA-seq, to answer these two questions. These are some field photos. Here we introduce FUNPOR pipeline, a long-read correction and annotation framework to correct the distinguishable frame shifts errors on nanopore reads and address the large data loss caused by the metagenomic assembly. For metagenomic datasets, neither assembly of nanopore reads alone nor hybrid assembly of nanopore reads with high-quality Illumina short reads could work well. In hybrid assembly, 67.4% of the nanopore data were unused. This is mainly because that the randomly distributed low-quality regions on nanopore reads were virtually all cut open by the assembly algorithm because these regions could not be mapped by any of the Illumina short reads. Besides, functional prediction accuracy based on post-correction nanopore reads was evaluated. Despite the large number of fake genes called by prodigal, functional predictions based on post-correction nanopore dataset showed high consistency. With the aid of the assembly-free long-read metagenomic approach, 42% more of the permafrost microbiome could be annotated at genus level, and the KO categories involved in nitrogen and methane metabolism were enlarged by 58%. Based on the FUNPOR pipeline, we firstly investigated the microbial community in thawed permafrost across different altitudes. 78.4% of the genera observed were shared in all permafrost samples at different altitudes. The abundance of shared genera accounted for more than 99% of the total annotated genera. Although the well-adapted core microbial community in permafrost were resistant to environmental changes, the strong light at the top and the high nitrate nitrogen content at the middle could lead to permafrost community differentiation. Then, we focused on the transcriptional activities of nitrogen and methane metabolism in thawed and frozen permafrost at HP 3500. The nitrogen metabolism in permafrost trend to be a closed cycle, and the increased activities of microbial denitrification was observed in the thawed permafrost. The active microbial community involved in nitrogen metabolism evidently shifted from copiotrophic microbes from proteobacteria in summer to oligotrophic microbes from actinobacteria in winter. The gas phase measurement of the thawed permafrost in the field showed a slow consumption rate of N2O. Meanwhile, obvious transcriptional activity of N2O reduction was observed in thawed permafrost, but not in frozen permafrost. These results suggested N2O was more likely to be consumed by microorganisms and prone to convert to N2 in thawed permafrost. This phenomenon can be further proved by our soil incubation experiment. For active methane oxidation, the active methanogenesis switched from hydrogenotrophic methanogens in winter to acetoclastic methanogens in summer, because the active acetogenesis facilitated by polysaccharide degradation in thawed permafrost. Besides, aerobic methane oxidation was the major methanotrophy process and was only detected in thawed permafrost. Based on transcriptional activity of key gene, the low methane production to consumption ratio of 1:38 was observed, suggesting microbial AMO can effectively transform CH4 to CO2. This speculation was consistent with the low CH4 emission rate observed in the gas phase on-site, while the CO2 emission rate was 50 times higher. Additionally, the incubated experiment also showed more CO2 was released with the thawing of permafrost. Finally, here are our main conclusions. iMeta, a journal to change the world.